Good morning, everyone. Yeah, actually, in our project, we are using the ASV, as mentioned by the previous speaker, and we would like to show the results that we have collected using the ASV and the optical sensor that we have done. Okay, I'd like to start with a short introduction to harmful algal bloom, in case there's any people who doesn't know. Algal blooms term is defined as an increase in algal cell density to several thousands of cells in the drop of seawaters, and which are induced by favorable environmental conditions, such as high level of nutrients, good sunlight, and the stratified water column. And when the blooms causes negative impact or harmful consequences to marine organisms and to humans, it is termed harmful algal blooms. And as you can see, some has discoloration, some kill fishes, and some impact other organisms like shellfish. Harmful algal blooms are basically divided or categorized into two types. The red type, which means it discolored the seawaters and caused by high density of phytoplankton species. And on the right-hand side, the toxic algal blooms, which cause shellfish or fish contamination with the algal toxin which they produce through the feeding. And red tide normally cause fish kill and shellfish death as well. And the toxic species normally are harmful to humans when we consume with these contaminated shellfish. In Singapore and Malaysia, we have many areas infected or affected by this kind of harmful species causing bloom in many areas. For example, late 2015 to early 2016, there's an extended bloom of a very toxic species in the northern part of Malaysia. And in Singapore, we have several fish kill since 2009. And we also have an extended dinoflagellate blooms, which lasted for about 3 months. And in Singapore, we mainly experience the red tide blooms as opposed to the toxic blooms which was observed in the eastern side of the peninsula of Malaysia. As we have experienced many types of blooms in our areas, so far we have 3 fish kills and we have a very big extended bloom, but it was confined to a very small area. And based on this, how are we going to monitor our algal blooms in our waters? And are they predictable? And as we know, blooms are very complex and they move around and they form blooms in patches. And to understand this, we need to understand the dynamics of the bloom formation, their behavior. And thinking of this line, we aim at using new tools, new advanced technologies for monitoring and detection of such bloom. And the project started with getting the kayak to work in the field and optimizing and for measurements of various environmental parameters, including the biological biomass. Our goals are to develop a sampling system using optical sensors, autonomous vehicles, advanced technique for monitoring the environmental changes and also the algal blooms movement patterns and the dynamics, and also to rapid detect, identify plankton species or bloom forming species using some of the techniques available, such as the some of the molecular tools, which we will show later. And first, I'd like to show some of the blooms which we mapped during the early 2016, and this was toxic dinoflagellate blooms which we found in this small little bay near the Silita Island. And we used the autonomous surface vehicles with optical sensors attached to the bottom of the kayak, we managed to map patches of high concentrated cells in the middle of the channel during the ebb tide about 9 a.m. And after a few hours, we came back to do the mapping again during the flood tide, and the bloom was basically gone. They were mainly pushed into the channel with only a few very small patches at the edge, in the middle of the channel. And for vertical profiling or distribution of the algal bloom, we use the optical sensors, and we measured from all the way from the inner channel to all the way to the mouth of the channel. As you can see from the left plot, during the ebb tide, we see concentrated patches which stretch downwards to about almost 2 meters, and at the outer part of the channel, the concentration was not very high, and during the flood tide, it's quite mixed, and the blooms extended down to more than 2 to 3 meters, and at average concentrations. From using this kind of methods, we were able to map the horizontal distribution and the vertical distribution of this kind of algal blooms, because sometimes those blooms are not found on the surface due to mixing and due to the movement of the current, and also tidal current as well. And this is the 5 days of bloom tracking and monitoring during the same bloom, which we observed early 2016. On the first day, they are all collected at the same time, different day, about 9 a.m. And on the first day, we could only detect very low concentrated patches around in the middle of the channel. And on the second day, we saw higher concentrated patches out here. And on the third day, 1st February, we saw a very concentrated patch in the middle of the channel. And on the last 2 days, the patches were all spread out and distributed within the channels. However, the high concentrated patches did not go out of this channel and spread along the Johor Strait. It was moving in and out of these small channels. And by using the kayak, we were able to map and study the movement of this bloom. And most of the movement are caused by the wind surface current and the tidal current exchange during the day, especially between the ebb tide and the flood tide. And early this year, we managed to map some of the features along this track that we have done using the optical sensor as well. We detected a diatom bloom. These are the good guys. They are not harmful. And we saw some pattern of accumulation. You can see from this left panel, this is a salinity plot, depth against the longitude here. And from this map, we were able to map what they call freshwater plume. We was from the terrestrial input, and the freshwater started with very high mixing, and it stretched all the way out actually to the mouth of the channel. And at the edge of the channel, we saw accumulation of diatom blooms with high concentrated subsurface depth about 1 meter, with a high concentration of about 20 microgram of per liter of chlorophyll, which is indicative index of the biomass of the diatom bloom. So from our transect, we were able to see these kind of features, like, for example, blooms hiding beneath the freshwater plume, and we were able to map the whole stretch of the water plume and see how this the ecosystem in this channel recover back to its original states after 5 days of sampling. And for detection of the bloom species, we used the real-time PCR, and we actually used and tested this system in the field by spiking with the some of the cultures and did the measurement in the field. And this method actually works quite well. It could identify the blooms forming species, and it is a very rapid detection method for target species, and it could be done in the field, and it could use simultaneously to detect several harmful or bloom forming species. And some of the results which we were able to conclude from the monitoring using such instruments that in the tropical areas, these toxic Caronia mikimotoi blooms at higher temperatures, and it also could form bloom with other species such as diatom, but they are the dominant species within the phytoplankton community. And actually they form blooms after heavy rainfall, and they could grow or multiply very fast with the surplus of nutrients such as the nitrogen sources which were transferred or introduced into the marine ecosystem through rainfall or through flushing and other means. And as compared to other regions such as species from Japan and Korea, similarly they bloom at higher temperature. They also could form or cause bloom with other species, and they like lower salinity, and they prefer surplus supply of nitrate. And temperate species tend to cause fish kill and shellfish mortality as well. We have not observed any fish kill caused by this species yet in our waters. And we also started the long-term monitoring at a monthly basis from January 2016. This is the total diatoms, which exclude the toxic species. And this is one of the toxic species that we are monitoring, the fish killer. These are the 2 fish killer species, and this is one of the toxic diatom species which we found this year, and at a relatively high concentration. We have never seen such high concentration before, and this toxic diatom actually is known to produce domic acid, which is very harmful to humans. And we were not able to capture the actual bloom based on the monthly resolutions. However, we did, during this bloom, we have the daily resolution as well. It captured quite well the bloom peaks and the high concentrated patches, but with the monthly resolution, we could not capture the very high cell bloom, very high cell density patches in the Silita areas. This slide shows the number of species that were documented and confirmed in the Western Pacific regions. And Malaysia has the highest number of Pseudonesia species documented and confirmed. And Vietnam, China, Malaysia is about 12 to 17. The rest of the regions only documented species less than 10. In Singapore, we have confirmed 5 species. And out of the 5, 3 of them are able to produce toxins. And these toxins normally known to cause amnesic shellfish poisoning, which is quite harmful to us. And so far, this shellfish poisoning has not been reported in Southeast Asia. And actually, these domoic acid producers are widely distributed in the tropical regions. Some of the trends of these newly found or documented species in our waters is that they are increasing in frequency and durations due to changes in climate patterns, also the nutrients regimes in the marine system, and they cause blooms in many areas around the world. And one example is that they formed the largest bloom along the west coast of the United States in 2015, and many of the beaches and shellfish were banned due to the contamination by the dominant acid. And some of these bloom cause death, and it could cause brain damage and some lung problem as well. And for monitoring, I think it's very important to include these species in our monitoring program in order to mitigate or prevent poisoning cases. And lastly, I'd like to summarize and conclude some of the work that we have done. As we have seen that the frequency of blooms associated with fish kill has increased in our waters, and due to the complexity of the blooms, sometimes we do find multi-species in the bloom, so it is quite difficult to mitigate or to predict them due to the multi-species blooms which we experience in our waters. And also the toxins which causes harm to other organisms, mammals and humans. And are we able to eradicate this kind of algal species? Currently, there's no single method that can be used to stop bloom. However, we could prevent new introduction. What we can do currently is to mitigate or to introduce control, prevention, to reduce some of the nutrients loads into our system. Lastly, I'd like to acknowledge the groups which helped or contributed to this project, especially these SENSEM engineers with their support, field support, and also TMSI staff, students, for their contribution to the project as well. Thank you.
So while Pablo sets up, are there any questions from the audience? Oh, yes, one.
Hi, Sandrick. Thanks for the talk. I noticed you said that you did qPCR in the field, and I was wondering if you might just say a couple more words on that. In particular, I was wondering if you found that it was efficient to do qPCR in the field, because the alternative would be to collect your samples and then take it back to the lab. And if so, what is the setup that you use? What is the piece of equipment? And is this something that you think is going to be part of like a in situ monitoring framework?
Oh, actually, we use the portable qPCR machine, which we borrowed from the one of the distributors. And why we use in the field, because sometimes there's a need to identify the species. If we could, for example, like the Pseudonychia species, right? Some are not toxic, some are toxic. So if we could confirm it is toxic in the field, we could take more samples for measurements in the lab. Yeah, that's one of the objectives that we have. And for using it as in monitoring programs, that's what you meant?
How long does it take to get your answer from sample to answer in the field?
About 2 hours, I think. Yeah. Including extraction, I think. Yeah. Or you can visit our poster if we have, yeah. And Li Ying will explain to you more, I think. Yeah. Thank you.
Any more questions?